All right, today we're going to dive into a completely new way of thinking about diagnostics. We're talking about a tool designed to take on one of humanity's oldest and deadliest foes, tuberculosis. But this is bigger than just one disease. It's really about a fundamental re-engineering of how we fight infectious diseases across the board.
So to really get a sense of the stakes, let's just start with the sheer scale of the problem. According to the World Health Organization, a lot of us think of tuberculosis as a thing of the past, right? Well, it's not. We're talking 10.6 million people affected every single year.
And tragically, of those 10.6 million people, more than 1 million die every year. That's what makes TB the world's single deadliest infectious disease. It's like this silent pandemic that just keeps going, claiming lives on an absolutely massive scale.
Now here's the thing. This crisis, it's been called a systemic moral catastrophe. And that's because it's not just about a tough bacterium. It's really a story about the failure of our tools. The fact that we can't quickly diagnose the drug-resistant strains, well, that's what's allowing the deadliest forms of TB to spread completely unchecked.
So you might be asking, what are we using right now? Well, the gold standard tools, like GeneXpert, they're powerful, for sure. But, and this is a big but, they have some pretty critical limitations, especially in the low-resource settings where TB hits the hardest. For starters, they need a constant cold chain. That means uninterrupted refrigeration, which is a huge challenge. They're stuck in centralized labs, and figuring out drug resistance? That can take days, sometimes even weeks. We clearly need something faster, tougher, and a whole lot smarter.
And that's exactly where this new idea comes in. We're not just talking about a small upgrade here. We're talking about a whole new category of diagnostics. It's called Intelligent Ambient Stable Molecular Diagnostics, or IASMD for short. And it's a system built on 3 really cool pillars of innovation.
Okay, pillar number 1 is what they call cognitive. Think of it as a super smart AI triage engine. What it does is pretty amazing. It takes chest X-ray images, fuses them with clinical data, and then predicts who is at the highest risk for the disease. It spits out a nomination score, which basically tells healthcare workers, hey, focus on this person. It's all about using those scarce testing resources with absolute precision. Next up, pillar number 2, invincible. And this one tackles that logistical nightmare we talked about, the cold chain. It uses something called extreme lyophilization engineering, which is basically a very advanced form of freeze drying. This makes the test's chemical reagents totally shelf-stable for a full year, even at 37 degrees Celsius. It just completely shatters that cold chain barrier, which means you can finally get these tests out to the most remote clinics on the planet.
And that brings us to the final pillar, decisive. This is the real core of the test. It's a dual orthogonal CRISPR system. Now, if you think of CRISPR as molecular scissors, this system uses 2 different kinds, Cas12a to look for the bacteria's DNA, and Cas13a to look for its RNA. Why both? Well, this double check not only confirms there's an active infection, but it also delivers a complete drug resistance profile. And it does all of this in just minutes.
Okay, so we've got these 3 pillars: cognitive, invincible, and decisive. But how do they all work together? Let's walk through the workflow and see how you get from a patient to a complete resistance profile in under 45 minutes. It's pretty incredible. It's really just a 4-step process. First, the AI does its thing and nominates a high-risk patient. Then, you take a simple sample, and it goes through a rapid lysis step. That just means breaking open the cells to get to the genetic material. Step 3, you add that to the 1-pod CRISPR reaction, and boom, finally, in less than 45 minutes, you have it. A complete, actionable resistance profile that tells the doctor exactly which drugs are going to work for that specific patient. So that all sounds great, right? But what does this new approach actually deliver in the real world? Let's talk numbers and break down the quantifiable leap in diagnostic power we're looking at. When you put them side by side, the comparison is just stark. Your time to result plummets from days or even weeks down to under 45 minutes. The cold chain, gone, completely eliminated. And the richness of the data you get is just profound. You're getting a resistance profile with the same detail as whole genome sequencing, which is like getting the bacteria's entire genetic blueprint, but you're getting it right there, at the point of care.
And the projected performance metrics are just as compelling. The AI engine is expected to have a predictive accuracy of over 0.95, that's measured as AUC, which basically means it's incredibly accurate at picking out the right patients. That intelligence alone is projected to cut the number of expensive molecular tests needed by 50%. And the CRISPR test itself? It shows over 98% concordance with gold standard sequencing.
In other words, it's getting the same answer, just way, way faster.
You know, when you step back and look at the whole system, you realize this is about way more than just a better TB test. By combining AI, this incredible reagent stabilization, and super-fast molecular profiling, ISDMD is really providing a blueprint for how we can respond to all sorts of future infectious disease threats and really bolster our global health security.
Of course, a vision this big isn't something you can tackle alone, and that's why this is being pursued through some really powerful partnerships. The project is actively building collaborations with leading research institutions like KOST, KACST, and Boston University. You really need to bring together all that interdisciplinary expertise to make something like this a reality. So if there's one thing to take away from all this, it's that we're looking at a system that can potentially stop the transmission of multi-drug-resistant TB in its tracks, simply by enabling immediate, targeted treatment. And that leaves us with a pretty huge question, doesn't it? Is this fusion of artificial intelligence and cutting-edge biology the blueprint for how we finally, for good, end drug-resistant epidemics?